One ordinary virus gave mice Parkinson's-like brain damage and months of movement problems. No toxins, no gene edits. So, can a simple infection spark Parkinson's-like disease, and what does that mean for us? Here's the plan. The problem with old Parkinson's models, what this team did differently, what they found, and what it honestly means. Parkinson's hits over 10 million people worldwide and destroys dopamine-making brain cells that smooth our movements. Most lab models force the disease with toxins or gene tweaks, but that may not reflect how it starts in real life. For decades, scientists have suspected infections and brain inflammation, mixed with our genes and environment, can help kick off Parkinson's. Viruses can cause different problems depending on who you are. Think Epstein-Barr and mono, or SARS-CoV-2 hitting lungs, heart, and brain. This team tried a new route. Infect mice with Theiler's murine encephalomyelitis virus, TMEV, a natural mouse virus. They compared 13 infected mice to 14 healthy controls to watch what changed over time. 1 week in, the virus was inside dopamine-making brain cells. By 1 month, those cells were destroyed where the virus struck. Dopamine-mimicking drug then produced a telltale movement pattern confirming dopamine neuron loss in the infected mice. Next, the pole test. Infected mice took longer to climb down, and they were still slower at week 20 when the study ended. They also ran a fancy treadmill that measures 100+ walking and balance details. The virus group showed Parkinson's-like gait changes. Put together, a simple infection led to both hallmarks, dopamine cell loss, and slower abnormal movement over weeks. Why this matters? It's a non-toxic, more natural model that could mirror how Parkinson's begins after infections in real life. Now, researchers can chase early warning signs, search for biological markers, and watch how the immune response reshapes the brain. The striking part: these deficits lasted months in mice after a single infection. Big caution: this is a mouse virus in mice. It does not prove that human viruses cause Parkinson's in people. But we can say it cleanly triggers Parkinson's-like damage without poisons or gene edits, and supports infections as one possible trigger among many. What we can't say, that city life, subway rides, or anyone cold raises risk. Those human questions are still open. Practical note: preventing serious infections is generally good for long-term health, but don't panic that every virus equals Parkinson's. Next up, the team will pit this model against older ones, look for early red flags, and map the brain's immune response over time. So, can a simple virus trigger Parkinson's-like disease in animals? Yes, this mouse model showed the brain damage and months-long movement issues without toxins or gene edits. What it means: scientists now have a more realistic tool to study how Parkinson's might start and how to stop it, while human causation remains unproven. With an aging world, cases will rise. Models like this may help us catch early signs sooner and protect more brains. Bottom line: in mice, a single viral infection reproduced the key brain damage and movement problems of Parkinson's. No toxins or gene edits, giving researchers a more lifelike model. For you, it strengthens the idea that infections can be one piece of the Parkinson's puzzle, not the whole picture. It does not prove everyday viruses cause Parkinson's in people, but it opens a smarter path to find early warnings and prevention strategies.